Okay, topic is molecular genetics. It's chapter number uh, 20 of our book, and today we will talk about the Western and Crick model of DNA structure. Whenever we talk about the DNA stands for deoxyribonucleic acid, or DNA is a molecule that carry genetic information. This was first discovered by Weston and Crick, which were awarded by the Nobel Prize in 1962 for discovering the double helix structure of the DNA. But work was started long before by others like Franklin's. Okay, what is a DNA? A molecule carrying genetic information. It is a small segment of DNA makes a gene. It consists of two parallel strands twisted together to form a double helix. A DNA molecule is wrapped around proteins to form a chromatin thread. During the cell division, the chromatin thread coiled tightly into a structure called chromosomes inside the nucleus. So, now we will talk in detail how is DNA organized inside your cells. During cell division, chromatin thread coiled tightly into structure called chromosome inside the cell nucleus. A molecule of DNA is wrapped around proteins to form a single chromatin thread. Each DNA molecule consists of two strands twisted around each other to form a double helix. A helix is a coiled structure like a corkscrew or spring. A small segment of DNA carries a gene that stores information used to make a single polypeptide. Polypeptides are used to make proteins, and proteins are responsible for determining the characteristics of an organism. So, in this way, we can say that DNA organized inside the cells. Now, we will discuss about the basic units of DNA. To understand how information is stored in DNA, we first need to know the basic units that make up its structure. The basic unit of DNA is called a nucleotide. Each nucleotide, which is shown here in the picture, is made up of a sugar called deoxyribose. In the center, we are having a deoxyribose sugar, a phosphate group, and a nitrogen containing base. There are four types of nitrogen containing bases in DNA. The bases are called adenine, represented by A, cytosine, guanine, and thymine. These may be represented by symbols as shown in the picture. The nitrogen containing bases, the deoxyribose sugar and phosphate groups can combine to form different nucleotide molecules as shown below. Here the first one is a adenine nucleotide, similarly thymine, guanine and cytosine. These are the four nucleotides. Nucleotides can join together to form long chains called polynucleotides. Each gene is made up of a sequence of nucleotide and the varying sequence result in different genes. For a gene made up of nucleotides, there are four different combinations of the nucleotides as shown above. It's a first one nucleotide adenine, second cytosine, third thymine, fourth guanine. Here it's a double helical structure of the Western and Crick model of DNA. It's a double helix structure. Okay. The DNA molecule is made up of two antiparallel polynucleotide chains. They are said to be antiparallel because the two chains run in opposite direction. The base of one chain are bounded to those of the opposite chain according to the rule of base pairing. There is a rule of a base pairing. Adenine, what is that rule? Adenine always bond with thymine. Cytosine always bond with guanine. Adenine and thymine are thus called complementary bases. Cytosine and guanine are also complementary to each other, and these complementary bases are joined together by the help of the hydrogen bond. Means these are joining by the help of the hydrogen bond. An uncoiled DNA molecule can be represented by the different base pairing. Here still it's a base pairing, it's a nitrogenous base pairing, how the adenine is doing the pairing by the help of the hydrogen bond with the thymine. In the same way how the guanine is doing the base pairing with the cytosine by the help of the hydrogen bond. So this picture is showing us the nitrogenous base pairing in the DNA between the four basic you can say nucleotides. There are the four basic nucleotides, and this thing is showing us the pairing between the four basic nucleotides. Here again still this picture is showing us the you can say a pairing of the nucleotides. Okay, now we can say that an uncoiled DNA molecule can be represented by a simple model that is just shown here. Here we can come for an uncoiled DNA molecule resembles a ladder. The twist of the coiled DNA molecule resembles a spiral, staircase or double helix. Here in this picture we are having a simple ladder also, which are having the joining of the different patterns of the bases with each other, and then the other one is showing us the double helical structure. So I guess the idea about the DNA and its structure become very clear to you all. Stay safe and happy. Allah Hafiz.